In this tutorial, you will learn how to use gel electrophoresis to sort and compare samples of DNA. First, connect the electrodes to the gel bed. Click and drag the positive, red electrode to the front of the bed. Then click and drag the negative, black electrode to the back of the bed. Next, transfer the amplified DNA samples from the test tubes to the well of the gel bed. Pick up the pipette from the bench, position it straight over the test tube, and click to fill the pipette with the sample from the tube. The full pipette will lean to the left. Drag the pipette to well number 2 and click to transfer the sample. Make sure to always start with well number 2, as wells 1 and 10 have already been loaded with standard markers. The pipette will lean to the right when it is empty. Repeat this for the remaining DNA samples, moving from left to right across the gel. Underneath the lab bench, turn on the power supply. Adjust the duration of your experiment and the voltage if necessary. 140 volts and 15 minutes is optimal. Then toggle the off button to turn on the current. Advance the time by 15 minutes by clicking the up arrow on the laboratory clock. You will notice that the DNA samples will have moved through the gel and have separated into bands. After 15 minutes have elapsed, either in real time or by advancing the clock 15 minutes, the voltage will turn off and the samples will have traveled down the gel. In the gel electrophoresis window, click the UV button to see the marker dyes in the sample. This concludes this tutorial on using gel electrophoresis to sort and compare samples of DNA.